Hi, this is Miss Litton, and this is my wonderful Period 3 class. Say hi.
Hi.
All right, so the first thing I need to do is, I know this says Chapter 11, Mendelian Patterns of Inheritance, but when the new framework was released from the College Board this summer, they interjected a whole section that doesn't necessarily apply to this in any way, shape, or form, but I will make the connection for you, okay? I'll show you why they are introducing this material. The reason why I tell you this is if you go to look in your textbook for this as reference in this chapter 11, you will not see it there, okay? But I have included links, and in this presentation you will have everything, and some of it will actually be familiar to you. So the very start of this chapter, this bit of information about Mendel, really talks about common ancestry. And that's your connection, right? Because if we're looking at how traits are inherited, we're starting off now by saying we have, yes, traits are inherited, but we have all inherited these traits from a common, what? Ancestor. So that's why they put it in this part. So I think it is of value to say it. I'm just going to reiterate, you're just not going to find this information as in the first section of your textbook. You're just going to get this from me, okay? All right, so there's pieces of information, there's pieces of data that we need to look at and that you can't ignore that point to why we have a common ancestry. And so on your notes, go to the very first thing there on your notes. I want to fill that in. Genetics, heredity, and evolution. Shared, conserved, fundamental processes, and I'll talk to you about those, and features support the concept of common ancestry for all organisms. Common ancestry for all organisms. Youngest bio buddy, tell the oldest bio buddy what it says in red after that. Go ahead. It says much of the information below will be supported in future units. This is the big picture showing the connections between DNA and evolution, not in text, it's been added in. All right, so the next part, the processes that I'm going to show you, these provide evidence that all organisms, archaea, bacteria, and eukarya, and then oldest bio buddy, could you differentiate between those three domains real quickly? Go ahead. What do archaea and bacteria have in common? Prokaryotic. Okay, and they are what? Unicellular. Okay, and then eukarya, right? And we'll talk about the characteristics of that, but that idea is that you're a eukaryotic cell, yes? Whereas bacteria and archaea are both prokaryotic. Both extant and extinct. What do you think extant means? Living. Extinct is not living. There you go. Okay. Are linked by lines of descent from common ancestry. Lines of descent from common ancestry. So here's evidence one. DNA, RNA, and ribosomes. Who has it? All cells. Okay, so DNA, you're nucleic acid, right? And if you are a prokaryotic cell, your nucleic acid, what's the shape of it? Circular. It's circular. You could have additional DNA called what? Plasmids, okay? And whereas in eukaryotic cells, the DNA inside the nucleus is not circular, it is what? Linear. Is that the only DNA eukaryotic cells have? No. No. Where else do they have DNA? Boom. Mitochondria and chloroplast, and that gives evidence for something else. What does that provide evidence for? Gosh, my heart is smiling. Endosymbiotic hypothesis. Exactly. Okay? Now, check with your bio buddy and go, did you know that? All right. Now, here's the deal. DNA, your code book, you will find that in all living cells. This right here is a virus. Is it alive? No, viruses are not alive. We're going to study them in the future, okay? Viruses are not alive, but they have nucleic acids in them. It's either DNA or RNA. It's either single-stranded or double-stranded. DNA is how information is transferred through DNA. All cells have that. And the way they transfer it is they make use of ribosomes. Ribosomes, what do we call ribosomes? They are the what? Workbench for protein synthesis. DNA is the code telling you how to build the proteins Where are you going to build the proteins? Out on the ribosomes. Okay? Now, I'm going to show you two pictures. Not it. Decide if you're going to take the top picture or the bottom picture. But the first one to go is the one who explains the bottom picture. And then explain the top picture. Okay? Go ahead.
Oh, yeah. So transcription is the same thing. Like an mRNA. So you have double-stranded here, right? mRNA is just another
Okay. This that I'm showing you right here, we have a whole chapter or two just dedicated to that. So it must be a pretty big deal. Okay? So it's how we use that DNA to code for proteins, and then there's another whole chapter on the regulation of all of that. Let's start with the stuff that we're most familiar with first. Right here, DNA making more DNA, that is called what? Replication. When does that occur in the cell cycle? S stage of interphase. Why does that occur? You're going to make daughter cells? Yes, you're making more cells. Either what are the two ways we can make more cells? Through what? Mitosis or meiosis, right? Mitosis and meiosis. But either way, the reason why you would do DNA replication is because you're going to make more cells. We good on that? Now, how you live your life as a cell has to do with the next part, transcription and translation. Now, if you have something like the Mona Lisa, does everybody know what the Mona Lisa is? Artwork, right? Okay. How many Mona Lisas are there? One. Is it in a museum? Yes. Okay. Can you take it home? No. It stays locked up in the museum. Do you agree with that? Okay. Are there copies of the Mona Lisa? Does it appear in textbooks? Yes. The copies, that would be the mRNA. Now, could I take the original Mona Lisa and go, you know what? I have this idea for some art. Do you mind if I just take this Mona Lisa and cut it up a little bit? Is that going to happen? No. Right? But if I have a picture of the Mona Lisa, could I cut up and manipulate that? Yes. Could I create some different artwork? Yes. Okay? That's what mRNA is. Messenger RNA. It's a copy of the original Mona Lisa DNA. Could the Mona Lisa have mistakes in the painting? Yeah, absolutely. As it ages, will it have more mistakes? Yes, because it'll start to what? Deteriorate. Okay, just keep that in mind. So here I have this messenger. I have a copy of the Mona Lisa. I can mix and match that up any way I want to create my art, my proteins. The way that occurs, that transfer of information from DNA to RNA, that is called transcription. I'm going to teach you all about that. How you use the mRNA to build that protein, that is translation. You're translating it. Now, when you translate something, same language or different language? Different language, right? You know that I'm fluent in Spanish. I'm not, okay? But if I say hello in English, right? Could you write hello down and you still write English and you could write the word hello on your notes? Yes, that would be transcription. It is the same language, right? Just a spoken version versus a written version. But if I say hola, okay, I have translated it into a new language. When you do translation, you're taking a nucleic acid language and translating it into a protein language. All right so far? Okay. Now, when you look at this picture of DNA, let's start at the top. That is a model. That is a diagram modeling DNA. What are all those letters there? Nitrogenous bases. Yes? What is missing in this picture that I can't see? Nucleic, yes, sugars and phosphates. In this model, the sugars and phosphates are represented by what? A gray line. Can somebody make that the white light of learning because it's driving me nuts because it keeps flashing. I don't know where the other thing is. Yeah, the gray line is representing the alternating phosphates and sugars. That's the backbone. Now, what are these in between the letters? Do you see a pattern there? Who pairs with who? Do you see that? Who pairs every time with adenine? Thymine. Who pairs with guanine? Cytosine. Now all they did is put the letters there, but you know some are purines and some are pyrimidines. Do you remember that? Talk about that with your bio buddy. Who are the pyrimidines? Who are the purines? And how are purines and pyrimidines different from one another? How many rings do pyrimidines have? One. And purines have two. And you always have a purine binding with a pyrimidine. Okay? Now, how do we remember poor primitive pyrimidines? They only have one ring. Who are the purines? Adenine and guanine. So adenine is a purine. Who is the pyrimidine that it binds with? Thymine. And we can see that right here, yes? Now, what do these black lines represent in between those letters right there? Bonds. What kind of bonds, though, are they? Are they covalent bonds? Between the purine and the pyrimidine? Between A and T and G and C? Are those covalent bonds? No, they're not. Do they have them drawn like a covalent bond in this picture? Yes. What kind of bonds hold those together? Hydrogen bonds. How many hydrogen bonds between adenine and thymine? How many hydrogen bonds between guanine and cytosine? Discuss that. How many between adenine and thymine? 2. How many between guanine and cytosine? Have I already taught you this? Yes. In the 4 important organic molecules. Whenever you look at a model or a diagram like this, look to see how is this accurate, how is this inaccurate. If you can do that, that means you really understand it, right? That means you really understand it. If I hear the band play a song, if I don't know that song, how do I know if it's accurate or inaccurate, right? I have to know it well enough to make a comment about it, yes? Same thing. So don't just look at these things and start, I heard people say covalent bonds, you couldn't remember. You need to start reviewing that stuff, okay? Here's your DNA. The process of using that DNA and making a copy, that is transcription. We're going to have a whole chapter just talking about how is that controlled? How is it that which DNA is transcribed at what time, for how long, for how much? Okay, we will talk all about that. But when you do have a transcript, this mRNA, what does the M stand for? Thank you. Every 3 bases on this mRNA is called a codon. The codons, now how many options do I have for this one spot right here? 4. It could have been adenine, thymine, guanine, or, yeah, uracil instead of what? Instead of thymine, yeah, okay? Remember, in RNA, there is no thymine. Instead, there is uracil. Okay, this was learned when we learned the 4 important organic molecules. Do you remember that? So let's start going back and reviewing those, all right? So you have multiple options for each one of these spots in this codon. 4 different options here, 4 different options here, and 4 different options here. Do you agree? Okay, so, but this combination right here, AUG, will always code for methionine. That is an amino acid, always. That is the genetic code that is universal in what cells? All cells. All cells use DNA, RNA, proteins. All cells use this genetic code that I'm showing it to you right now. This will code for methionine. AUC, UCG. This one, though, is not coding for an amino acid. What does it say? Stop. That must mean we're done building that particular protein. Don't add anything on there. That is like a stop sign. This will give you your polypeptide. Going from DNA to RNA, transcription. RNA to a protein, translation. We're taking a nucleic acid language and changing it into a protein language. I'm thinking of a number between 1 and 5. Tell your bio buddy your number.
1.
My number is 5 because that's what I want you to earn on the AP exam. Whoever is closest to 5, pass or play, give a summary of this page right here. Go ahead. All right, now come back to me again. Do you remember when we looked here and I told you viruses are not alive? But they do have, there's two things they always are going to have, some sort of nucleic acid and some sort of protein coat. They may or may not have an envelope. Not all viruses have an envelope, and they're not all in this shape right here. But they all have a nucleic acid and they all have a protein capsid. Now, what HIV, what is that? Do you remember? I mean, just in your common vernacular, do you know what they call that? What type of virus they call that? It does attack our autoimmune disease. Yes, autoimmune disease. It attacks our immune system. Do you have another name for it? Anything? What does HIV, do you know what kind of virus it is? What kind of nucleic acid it has? It is an STD, but it can be spread in other ways as well. It's an RNA virus. It has the ability to take its RNA, and it has an enzyme that is packaged with it called reverse transcriptase, and it'll take the RNA message and make it into a single-stranded DNA message. And then it has another enzyme called replicase who makes it double-stranded. And this is how it is able to take its RNA message and insert it into our own cells. Because the code is universal, and it can insert its code into you, just like if you could get a what kind of virus? Computer virus. Because it's all the same code, and so it can insert its code into your computer and then wreck your computer, yes? And whole systems of operations. So on your notes, go to your notes, and go, DNA and RNA are carriers of genetic information. The proper storage and transfer of this information are critical for life to continue at the cell, organism, and species levels. Non-eukaryotic chromosomes are what shape? Circular. Good. Are circular. Eukaryotic chromosomes have multiple linear chromosomes. What is the workbench for protein synthesis? Ribosomes are found in all forms of life. Viruses are either RNA or they are DNA viruses. DNA is interchangeable between living organisms. It can be moved from one organism to another. That's why when we do our genetic engineering lab, we're going to take this plasmids, we're going to manipulate plasmids and put eukaryotic genes into plasmids for the mutant red fluorescent protein is what it codes for, and then we're going to make bacteria code for that protein because it is interchangeable. But this is kind of a misstatement because when it says DNA is interchangeable between living organisms, non-living organisms can insert their genetic code into host cells as well. Okay, so that's the first point that we want to make about common ancestry. The second one, okay, is about a universal genetic code. A, T, G, C, and U are the nitrogenous bases found in all living organisms. The central dogma of genetics states that in all life, it means it's conserved, DNA replication ensures the continuity of hereditary information. You're passing on your DNA, you're passing on your genes. Genetic information flows from nucleotides in a gene to a sequence of amino acids in a protein. And that's through transcription and translation. Now, do you remember how I said right here, AUG codes for methionine? Remember that part? Okay, they have one right here. Here it is. Universal genetic code used by all cells. You can look up any possible sequence of 3 nitrogenous bases on that mRNA, and you will know exactly what amino acid it codes for. So when you look right here, AUG codes for methionine. Now, I want to make it a little bit bigger here and point out a couple of things. So the first letter is here. This is the way you would read it. And then the second letter and then the third letter is over on the right. Look right here at leucine. How many different ways are shown that you can code for leucine?
4.
Yeah, 4, but look right above it. Do you see right here? Here are 2 other ways to code for leucine. Remember, at each one of those spots, you have 4 different options that each one could be. So you have like 64 different combinations of 3-letter words that you could make. And how many amino acids are there? 20. So there's going to be some redundancy. Do you agree with that? Okay. However, there is only one way to code for methionine, and that is AUG. I'm going to tell you something else. It is the first amino acid in all proteins. How many proteins? All. Same one. Now, do you remember how we talked about folding proteins into their right shape? Remember chaperone proteins? Remember that whole deal? Okay. In the process, it's not only just folding. Sometimes they snip things out as well in order to form something that you need to form. The same gene, remember the Mona Lisa? If I make a copy of the Mona Lisa in the mRNA, I could cut and splice that mRNA in different ways to create different art. Do you agree with that? Okay, so that's the same thing here. When you go back and look at the DNA going to the RNA, you can splice the mRNA any which way you want. So from one gene, you could get multiple proteins from just one gene, depending on how you modify it. Are you all right with that? Okay. Now, look on here and try to find how many stop protein, or how many stop codons are there. Look for stop codons. How many different stop codons are there? How many do you think? 3. Okay. So we have 1, 2, 3. So there are 3 stops. These are like periods at the end of your sentence. Okay, and again, we're going to spend a whole chapter just kind of looking into that and exploring how that works and how, let's say there was a mistake. Let's say instead of CUG, okay, instead of CUG, we had CUC. Would that be a problem? CUG and here's CUC, they still code for leucine, right? That is what's called a silent mutation, because we have the wrong nitrogenous base, but it didn't change what amino acid we had. If we had, though, instead of UUA, we had UUC, the first 2 are the same, and now the third letter is different. Would that cause a difference between UUA and UUC? Yeah. You have a different amino acid. If you have a different amino acid, do you remember when we studied the amino acids and we looked at, when we say there's 20 different amino acids, what's the part that varied? The R groups. So if the R group varies, if it goes from being polar to non-polar, or if it goes from being non-charged to charged, right? Could that affect the folding pattern? And then that could affect whether or not the protein is in the right shape, right? Sickle cell disease. What's the wrong with sickle cell disease? Do you know? The shape of the red blood cell. And why is the shape wrong? Because you have the wrong amino acid. Why do you have the wrong amino acid? Because you have the wrong mRNA. Why do you have the wrong mRNA? Because you have the wrong DNA. A mutation in the DNA means the copy that you have is going to be wrong, right? Because the original is wrong. That means the protein that you're going to code for is wrong. The proteins that you make is called the phenotype. What's in your DNA is your, anybody know? Genotype. And that's what we're going to be talking about in this chapter with Mendel, is what did the gene say? How does that play itself out in the phenotype? Then next unit, next semester in January, we're going to get into the details of how this takes place. And we'll back it up to that point. Okay, not it. Tell them one new thing you learned about this.
This kind of feels like the mutations, kind of like why you don't copy homework. If it's wrong, then you're screwed. The moral story don't copy. But interesting fact is mRNA can represent the same amino acid, even though it might have different components.
Okay, so let's add this to our notes. Okay, we have already, look at little number 2 under B2. Genetic information flows from nucleotides in a gene to a sequence of amino acids in a protein through transcription and translation. Oldest bio buddy, could you differentiate between those two words, transcription and translation, please? What's the difference between those two words? Transcription is DNA to RNA. Still nucleic acid though, right? Just a different kind of nucleic acid. Translation is using the RNA to build a protein. Okay? Very little of your DNA is actually translated into proteins. Just a little over 1% of all your DNA is translated into proteins. Very small amount. We'll talk more about that in a little bit. So, then number 3, phenotypes are determined through protein activities. Phenotypes are determined through protein activities. Okay? So number 1 evidence, DNA, RNA, ribosomes. Number 2 evidence is your central dogma. Okay, this genetic code. Yes?
Can there be changes in the DNA sequence during the transcription that would affect, like,
yes, usually mutations. However, it is perfectly normal to change the mRNA before you go from a protein. Do you see what I'm saying? There would be some mistake between DNA and mRNA, but once you have the mRNA, you can that's where you get the altering done. The copy is what you alter, not the original.
Okay.
Does that make sense? Yeah. Alter the copy, not the original. Now, what kind of things can alter the original? UV radiation, carcinogens, right? Toxins, your diet, hormone influence, all those kinds of things could actually influence your DNA. Your DNA then codes for your RNA. It could be messed up then, right? AKA cancer, right? If your DNA is messed up, you're going to have the wrong mRNA, you're going to have the wrong protein. Remember, RAS is like stepping on the make a bad rats, right? And it'll say go even when it should say stop. All right, good. Now, well, bad, but okay, number 3 is our universal metabolic pathways. Now, it could be direct metabolic pathways like cellular respiration. What's the first step? What's the first step? Glycolysis. Who does it? All cells. Who? All cells. Okay? So that is a metabolic pathway that is universal that points to a common ancestor. Okay? Whether or not you have mitochondria or whether you're doing aerobic versus anaerobic respiration, following that is a universal pathway. There are other pathways as well. Remember we talked about cell signaling. Not it. Pick the picture on the left or the picture on the right. Left, right. Decide which one you're going to explain. Should be no problem for you.
Single transition but different ways. One receptor receives two different pathways. We got 2 receptors and 1 pathway. 2, and then we have inter-crossing pathways and stuff like that.
That was last unit, right? That was last unit. Is talking about cell communication. That cell communication is done by all cells. It doesn't matter if you're if you're unicellular or what? Multicellular. This still moves forward. This still happens in all life. So let's write this one down. Maybe metabolic pathways one. ATP, energy, glycolysis in either aerobic or anaerobic respiration to make ATP. Cell communication in what kind of organisms? All organisms. Involves transduction of a stimulatory or inhibitory signal from cells organisms in the environment. Single-celled organisms, signal transduction pathways influence how the cell responds to its environment. Then I gave you a couple things there, quorum sensing and chemotaxis. I believe quorum sensing, you watched a video on it, right? TED Talk. Now, that one's kind of interesting to me, okay? Because this is a unicellular organism, but they can work as a what? Group. Okay? Foreshadowing, maybe. Okay? So, oldest bio buddy, take this one right here, and then I'll review it with you. But is it oldest bio buddy's turn or young? Yeah. Young, go young, go young. I like, go young. Right, I'm sure you could think of a lot of analogies for that, right? In Star Wars or something, and the clones, right? And how all are controlled by one hub or something, but that's exactly what it is, in the sense that, do you guys not watch Star Wars? Is that an okay Karen kind of thing? No. See, did I use it correctly?
Okay.
Okay. All right, Karen. Yeah. Now, come back to
you combine like two different things.
Oh man! How could have I? Should have I said it?
So like,
all right,
like the idea of Karen by itself, it's just like Karen, right? And then like you kind of did like okay boomer. Like,
oh, I did. I did.
So you went like okay boomer and Karen, and you went okay Karen.
You should make okay Karen.
Thank you. All right.
Hey, make it to go back.
Now, okay, Karen. All right, now, on this one bacterium is producing a chemical. This is the synthesis part. It's producing a chemical that it has a what for? And it can not only receive what it's making, but what others are making. So that tells them when to move into the next stage of whatever they're going to do. So they can work as a unit. Okay? Chemotaxis is the other thing on there. Chemotaxis is just moving towards or away from some sort of chemical stimulus. Towards or away, from food, or towards or away from some chemical in the environment that it doesn't like. That's all chemotaxis is. Okay, multi-celled organisms, signal transduction pathways coordinates the activities within individuals that support the function of the organism. The function of the organism as a whole, like temperature determination. We're going to talk later about that. We are used to sex determination being just genes. If you're XX, you would be what? Female. XY, you would be male in a binary world, just in the biology of it. But I think I've told you this before. You could have somebody whose genotype gene says XY, but they present phenotypically as a female. Okay? Why is that? Because the Y chromosome could have been masked. And so its proteins were not expressed. And so we default, in our mammalian way, we default into female if there's no Y. Does that make sense to you? Okay? So you can have a masking of some sort. Now, other organisms, fish, amphibians, it could be temperature dependent. If it's a really hot or a really cold temperature, you might be female. And if it's a moderate temperature, you might be male. Okay, so that is determining which genes are expressed. That's where your environment is influencing what your phenotype is. So there is that aspect to it. We will get to that. What we usually think about in multicellular organisms is always what? Homeostasis, right? And it could be through local signaling. It could be through two neurons talking to one another. It could be hormones from one gland to target tissue in your body. It could be pheromones. It could be chemical signaling between two different organisms, but usually it has something about maintaining homeostasis. So Are metabolic reactions like glycolysis, either followed up by aerobic or anaerobic respiration, all of our cell signaling. This is universal, again, pointing to that common ancestor. Okay, so the big ticket here they want you to have in your head, which I think you would understand it now, not it. Go ahead and explain that right here. So if on the AP exam you had an essay where they gave you a prompt, shared conserved fundamental processes and features support the concept of common ancestry for all organisms. Please describe 2 processes and how they point to common ancestry. You see what I'm saying? That would be an essay prompt where you would use one of these 3 in order to explain them. Okay? Now let's go on and move not from just all life, but let's focus on right here in this. There's archaea, there's bacteria. Who's right here in the middle? Eukaryotes. So eukaryotes have some things that are unique to them that point to their common ancestry. Whoever explained this one, other bio buddy, you explain this one. Go ahead.
Oh, eukaryotes.
I'm trying to block that.
Linear chromosomes, and they're all traits and stuff that like eukaryotes all have these traits. The bottom right.
All right. So, number 2, the existence of these properties in organisms today implies they were present in universal ancestor, and that present life evolved from a universal ancestor. In eukaryotes, conserved core elements provide evidence for evolution. Presence of a cytoskeleton, a nucleus, membrane-bound organelles, linear chromosomes, and the endomembrane systems. Who's part of the endomembrane system? Reticulum, rough and smooth, Golgi apparatus. That's part of your endomembrane system, right? Okay, good. So now what our task is, here, read this. Go ahead.
I don't want to do that. All right.
Now, think about the significance of that. If it would take you 57 years to take one of your cells and identify the sequence of bases within it, 57 years without stopping, okay? Yet, each time a cell replicates itself, it makes an exact copy of that 57 years worth of reciting information during the S stage of interphase. Do you agree with me on that? And then during mitosis, organizes it so that that 57 years of reading is in this one and 57 years of reading is in this cell. Does that kind of blow you away just a little bit to think about how magnificent each of you are and that each of your cells do this all the time without making too many mistakes? You all look pretty good to me. Okay? So what our job is going to be in this last chapter is to talk about how do we pass this DNA down? What could we predict if we know who our parents are? What should the offspring be like? That's where we're headed. And to do that, we're going to talk about Mendel. Okay? And I'm going to stop right there. 39 minutes of fun. Hope you're making good choices.